Good morning, Liz.
Good morning. How's it going?
Pretty good. You guys had any cold weather yet?
Oh, we get a little frost in the mornings.
Yeah.
Not properly cold yet, though.
Yeah.
I don't know. It was pretty, like last week. It was pretty cold.
How are things out west?
Wet today. I had a short walk to my office this morning, which I was fortunate. I got here early enough to get a closed parking place, so it's good.
That's key. All right, I'm just renaming myself because somehow I am. Oh, I'm Waterbirds because I'm a host, maybe. Okay, co-moderator.
Yeah.
And then I think what I found, just having done the back of the house moderation for the career panel, is that it's helpful if I'm the host so that I can be in the back making other people co-host and you can focus on talking to people.
That sounds great. Do you want to just grab that?
Yeah, if that's cool with you.
Oh, yeah. Yeah.
Oh, yeah. Here we go. Let's see. Tom, I can say I would like to. I will figure this out. I would reclaim the host. Okay.
That looks like it worked. I don't think I needed to do anything.
Yes.
Okay.
Okay. And so I don't know how you'd prefer it. I would be fine to just make all the speakers co-hosts as they come in so that I don't have to be shuffling. And then the worst thing that happens is, you know, all the speakers will have slightly more power than they would otherwise. But I think people know what they're doing.
Yeah, that sounds good.
Is that okay? I'll just get to work on it now, just in case anything falls apart on my end, which it could at any moment.
Can I just test my slides and then hop out so I could go watch the other?
Yes.
The other session.
And while you're here, I'm also going to make you a co-host, which I think.
Yeah, but then I'm going to leave though.
I think it might stay. We'll see.
Okay.
But yeah, so you are now. You should be able to screen share, and I'm going to let. Oh, Don just let them in. Great.
Let me see. Do you see? Hold on.
Hello.
Hello.
Can you see?
Hello.
Hi, we're just in the middle of doing some. Oh, hello. We're just testing out some screen share. Oh, that looks
slides.
That looks better. Yeah, it was really zoomed in to begin with, but now it looks like it's filling the screen appropriately.
Okay, great.
Looks good.
Cool. I'm gonna go watch the other talk and then I'll be back.
Okay, we'll see you back here.
Okay, bye.
Cool.
All right. So I'm sorry, you'll have to forgive me. Is your name pronounced Mateus?
Yes, Matthew. It could be Matthew.
Whatever you prefer.
Okay, can I share the screen?
Give me one moment. I'm going to make you a co-host and you should have the ability. Okay, you can give it a try now.
Okay. Okay, it is okay now.
Well, so we can see your screen. I wonder if there's a lag because I can tell we're still looking at the multi-slide view, not the presenter view.
Now it's okay. No.
I'm not sure. Maybe I don't know. Don, what are you seeing?
I'm still seeing the edit mode, not the slideshow mode.
Do you want to try switching slides and see what happens? I can't tell if it's frozen or if it's just the wrong view.
And now?
It's exactly the same. Maybe stop sharing and try again because it kind of looks like it's frozen.
Okay.
Try one more time just in case. Okay.
now?
Okay. Oh, something different is happening. We're seeing a blank screen. Maybe can you try advancing your slide again?
Now I change the slides, okay?
Hmm. We're seeing a screen share of a blank screen now,
Okay.
like black.
Mm,
Oh,
maybe,
okay.
uh,
Seems like it doesn't matter how many times you do this. It's always, always hard and always.
Yeah.
I feel like Zoom still surprises me in a bad way, at least once a week.
Mhm.
And
Okay.
now?
Oh, yes, you've done it.
Perfect.
Okay, but because I have to share not the presentation, but the screen.
Oh, okay.
So I try again, okay?
Yeah, we have 15 minutes, right,
Yeah.
to figure this out?
Okay, so now not the
Take your
presentation,
time.
but the screen. It is okay, yes?
Yes.
Okay.
Do you want to try advancing just to make, yeah, that looks good. Does that work on your end?
And you can see only the presentation, no additional.
That's correct.
yes?
Yes.
Okay, so thank you and see you.
Okay. Okay, good.
And stop sharing. Okay. Thank you.
Excellent. Thank you. Could you share your pronunciation of your name again, just so I come close? How do you say your name?
You can say Matthew in English or Mateusz.
Mateus.
Okay, it could be Mateusz.
Mateus. Okay, I'll do my best.
But you should say szy na koń, szy at the end.
Mateusz?
Yes, the better.
Mateus.
Yes. Okay, thank you.
And your last name? How is that?
Ledwoni.
Oh, that's harder. Ledvon?
Oh, yes, okay. It's right. It's right. Okay.
Okay, well, apologies in advance, and thank you for your mastery of English.
No problem. Thank you very much.
Yeah.
All right. Hi, folks. I think I've just made you each co-hosts. So we should be able to do any testing that you want to do, whatever you need to feel comfortable. You'll be able to share your screen at this point, I believe. I'm Liz, by the way.
Nice to meet you.
Just try to share my screen really quickly just to make sure it's working. Is that good?
It looks great. Yeah, Don will be doing all the introductions and I'll be doing the background tech. So if anything, if you need anything, you can message me privately if things are not going well.
Well, I'm also just gonna share my screen real quick, make sure it's going through. Okay, cool.
Do you want to check and make sure that the screen view works? Just to see, we saw your
Like that's full sized.
Mm-hmm. Just.
Is that's only showing part of it, right?
Yeah.
Interesting.
It's always a surprise what it will do. So, okay.
Yeah, it's just weird. I haven't had that issue before. Hold on.
Okay. Oh, that looks good.
Okay, cool.
Are you working with Adrienne Kovach?
Yeah, she's on my committee. I work with Sharp.
Oh, cool. I'm at UNH, so I've had a chance to do some minor collaborations with her.
Oh, cool. I
work
was
as
just
well,
up
but-
there.
Oh, really?
2 months ago. Yeah.
Oh,
I
gosh,
was finishing my lab work.
cool.
Lisa, am I pronouncing your name correctly? Would you prefer Lisa? What would you like me to use?
You can just say Lisa. It's
Lisa?
Elisa, but
Okay.
Lisa is good. Lisa Elisa.
Okay.
Mhm.
Okay. Thank you.
Don, I wanted to let you know that I've gone through all of your edits from the migration chapter, which is what I'm presenting. And so they're really helpful, first of all. But I was able to find like a lot of the things you suggested was to come at the same question like a different way. And it was interesting. I found almost the same results that I had found the first way, doing it like your way as well, which has been really nice to add into the paper to add some like robustness. Like, for example, like one of your comments pertain to like our estimation of like 74% of pelicans like migrate. And so I did it the way that you had suggested, and I came up exactly at 74% as well.
Oh.
Kind of funny.
Well, that's good. I mean, that's really good. I mean, when we come up with those self-definitions, I guess you could call them, they often get questioned. So if you've done it a couple ways and the take home is the same, that's great.
Well, I also wanted to say I'm going to talk about it a little bit in this presentation as well, but I put in a 7-day lagged rolling average for sea surface temperature and reran the model. And the model performed worse, which is whatever, but the relationship stayed the same of increasing SST even with a 7-day average rolling lag. So I might look at like a 14-day or maybe like a 21-day and see how far back I need to go to like potentially get a negative relationship there. But I at least have like the code figured out for, like I said, sort of averaging that time length. But I thought that was interesting that even with a 7-day rolling average lag, it was a worse model and I had the same relationship, which I thought was, like I said, that I wasn't expecting I was expecting a 7-day average rolling lag to have a negative sea surface temperature relationship instead of the positive one. But
right. That is a little surprising to me too. I would have thought that was enough lag to see a change, but yeah.
Anyway, like I say, I that was the last one because it involved the most coding that I like needed to do. So I just finished it up like a couple days ago. And like I said, I'd like to go back and explore more. I might try to do like a pseudo sensitivity analysis and see where exactly the change is from something that's negative to positive. So like I might put in like 21 days, and if it's negative, then do like 18 or 14 and see if I can plot where that like switch occurs. I think that could be informative. But
yeah, I like that thought. Yeah. Brad, when do you switch? When do you move?
I moved last week, so I've been at Duke for a week now.
Oh, okay. Yeah.
Yeah, I'm in DC right this moment, but I've been living in Duke, or I've been living in Durham for the past like 10 days. Like I said, I just had my first like official work week passed. So.
Yeah. Wow. You're juggling quite a bit then. Still trying to get manuscripts finished up and out. Yeah.
Yeah. It's not too bad. I'm excited. This is my favorite chapter to talk about. So I'm always really excited to talk about the migration stuff. But the oil spill one has been a little bit more of a slog going through like rewriting a lot of it just for Pat and I really appreciated the kind of different perspective that you brought about like instead of highlighting the results of the model necessarily, just talking more about like the experience using GNOME. And so that's been helpful. But like I said, it's involved the most like writing and kind of thinking as well. But
yeah. Do you guys have an outlet in mind for that yet? I can't remember if you had somewhere you were targeting. Yeah.
I mean, initially we talked about like marine pollution bulletin or something like that. But I think if we can make it more expansive, we could actually send it to a wildlife journal, which at that point opens it up quite a bit. So Pat and I still need to have that conversation.
Yeah.
So I'll keep an eye out for our final presenter to give them co-host abilities, but otherwise I think we're good to go. That presenter is going fourth, Akshita? Yeah, okay.
Yeah. We got the email from her this morning or overnight. So yeah. Liz, would you want to test your ability to share screen?
Well, that's smart.
Just in case.
Yep. So I'm gonna pull up Mateusz's just in case. See how that goes. Why would it want me to leave the meeting? I certainly don't want to do that. Okay, so what are you guys seeing?
Just the slide. Looks great.
Okay, great.
Yeah, full screen time.
And I have all of everybody's slides on my computer, so if anything does go wrong, I can just advance through slides for folks. And I know these two sessions are really back to back, so my guess is that we'll get a flood of people as soon as the previous session's last talk is up. And I can keep working on the admit if that helps.
Liz, I have a slide I could share just as a backdrop as people join.
I believe I've also made you a co-host, so you should be able to do that too.
How did that work?
Looks great.
Good. I see we have over 30 participants. We're a couple minutes past the hour, so we should probably begin to keep us on track. My name's Don Lyons. I'm very ably assisted in moderation by Liz Craig this morning. And I'd like to welcome you to the general session that will go on for the next 2 hours in this Zoom room. And we have a really nice slate of speakers. I'd encourage everyone to stay on mute if you can while the presenters are giving their talks. Feel free to enter questions in the chat at any time, and we can refer those to the speaker, or at the end of the talks, we can work on a process of people unmuting and asking questions. If that gets unwieldy, we'll switch to raising hands to ask questions. So to begin our session, I'm going to first quit sharing my screen, and I'd like to introduce Matusz Ledvan, who is coming to us from Poland, I believe, and is going to talk to us this morning about Whiskered Terns. So please, Matusz, take it away.
Okay, thank you. Hello, everyone. Now I try to share my screen. Can you see my screen? Yes?
That is excellent. Please proceed.
Okay, thank you. Hello, everyone. Thank you for introduction. My name is Mateusz Redwoń, and I work at the Institute of Systematics and Evolution of Animals, Polish Academy of Sciences in Kraków, in Poland. Co-authors of this work are Adam Flis from Institute of Nature Protection, Agata Banach from my institute, Grzegorz Neubauer from University of Wrocław, and Frédéric Angelier from French National Center for Scientific Research. I would like to present work titled "The Baseline and Stress-Induced Prolactin and Corticosterone Levels in a Species with Female Offspring Desertion," the case of Whiskered Tern Chlidonias hybrida. In the majority of bird species, both parents are involved in parental care, and conflict occurs between male and female because each one tries to maximize its own fitness. So it is expectations and it is real life. And one way of increasing one's own reproductive success is to desert a partner and brood, leaving the other one to care for the offspring. And natural, not caused by harsh condition offspring desertion by a single parent, is common in many bird species, especially in the caladiformes, particularly in shorebirds. As for the Whiskered Tern, Whiskered Tern is a marsh tern. It nests colonially. It is a socially monogamous tern. In this species, both mates incubate and brood the chicks. However, females feed chicks less than males, about 25% less than males. Among terns and gulls, desertion during chicks rearing period has been described only in this species. However, in common tern, it has been shown that females abandon parental care sooner than males, and it is happening during post-fledging period. In Whiskered Tern, almost all females desert. 97% of deserters are females, and half of the females desert during pre-fledging period here, and the other half during post-fledging period. Desertion starts when chicks are terminally independent, when they are about 5 days old. In the picture, you can see percentage of brood deserted by Whiskered Tern by females during subsequent weeks of chicks' lives, and here during the first week, only few females desert. After female desertion, males increase provisioning rates to compensate for the lack of the female, and males continue parental care for at least 1-2 months after female desertion. And female desertion has no effect on fledging success. So in this species, desertion is a natural process, natural behavior, and female desertion has no effect, no negative effect on breeding success. Two hormones are especially involved in regulation of parental care, corticosterone and prolactin. First hormone, corticosterone, is involved in the decision to abandon reproduction in response to stress. And for example, experimentally elevated levels of corticosterone are associated with reduction of parental care and with induced nest desertion. So corticosterone is a stress hormone. In contrast, prolactin is a hormone generally involved in parental behavior, in incubation, brooding, and food provisioning. And several studies have reported that reduced prolactin levels can be associated with reduced parental care. Until now, prolactin and corticosterone levels in species with uniparental desertion have been studied only in 3 species. In Kentish plover, here in semi-palmated sandpiper and little oak, and nothing is known about Whiskered Tern. So the main aim of this study was to answer the question: do males and females differ in baseline stress-induced hormone levels in this species? We predicted that females should have lower baseline and stress-induced prolactin levels and higher baseline and stress-induced corticosterone levels than males, because they are more likely to desert than males. Assuming that decrease in prolactin in response to a stressor is a surrogate measure of parental investment, we predicted that females that deserted their chicks after prolonged stress would have lower stress-induced prolactin levels compared with non-deserting birds, with non-deserting females, and with males. Study site. We carried out fieldwork in 2016 and 2017 on carp ponds near Zator in southern Poland. Annually from about 600 to 2,000 pairs are breeding there. Carps are shallow and covered mainly by fingered water lily here in this picture, and by water chestnut. And these fish ponds are the most beautiful I have ever seen. We had erected plastic mesh fences around the nest with eggs here to prevent the chicks from escaping until they fledged. We usually enters the colony twice a week to study breeding success and breeding behavior. Adults were trapped by roof trap. Here you can see the roof with the two holes, and birds quickly fly into the trap through the hole, and success in bird trapping was very high. It was about 90%. Adults were caught during chicks rearing periods before female desertion, so between the hatching and the 7th day of chick's life. We had to run or swim as quick as possible to take the blood within 3 minutes after capture to obtain baseline hormone levels. So we had to be like this guy from film Crouching Tiger, Hidden Dragon. We have to be very, very fast. And after capture, bird was placed in the cloth bag. After 30 minutes, the second blood was taken to measure stress-induced hormone levels after acute stress. And Frédéric Angelier from France and his team analyzed hormone levels. In 2017, we conducted a removal experiment. Adults were caught and kept in captivity for 24 hours to study the effect of prolonged stress on hormone levels and brood desertion. Therefore, this removal experiment was conducted when chicks were older and no longer required intense brooding. After 24 hours after trapping, we collected blood samples and birds were released. We carried out behavioral observations of marked birds. You can see bird marked with hair dye here to determine whether the birds returned to the nest after release and to estimate breeding success. We used linear models, GLM, and depending on the analysis, various factors were taken into account in the model. We usually included in the model sex, chick's age, body condition, and body condition was connected with body mass. We also included in the model interaction of these effects, but it depended on the model. And the best models were averaged, and model averaged prediction were applied. And results. I show only the most important results. Here in the picture, we have males, females, baseline, and prolactin level after acute stress after 30 minutes after capture. Prolactin level both in males and females decreased after acute stress. Only in 3 birds increased. The level of corticosterone increased after acute stress in all 41 birds. The baseline and the most important results, the baseline prolactin level here on in these 2 pictures decreased similarly in both sexes and both years in relation to chicks' age. However, there was no difference between males and females. The stress-induced prolactin level here in the bottom, this stress-induced level decreased in relation to chicks' age in both sexes, and the most important, these levels were higher in males than in females. Stress induced corticosterone levels after 24 hours after removal experiment, after 24 hours in captivity, was negatively correlated with body condition in females, but not in males. So females with low body condition here were more sensitive to a stressor. None of 9 males released after removal experiment, after being held in captivity for 24 hours, deserted, whereas 4 females, 29% of the 14 females kept in captivity deserted. And this difference was statistically significant. So more males than females return to the nest, which indicate that males are less sensitive to the stressor. And there were no statistically significant difference in hormone levels between males. Here, females that deserted, females that didn't deserted, and females that deserted. However, females that deserted had the lowest prolactin level, and this lack of statistical significance is probably due to lack of statistical power. And to sum up, a summary, we didn't find difference in baseline levels of prolactin and corticosterone between the sexes. The decrease in both baseline and stress-induced prolactin levels in relation to brood age suggests that the differences between the sexes in hormone levels could occur shortly before desertion. So desertion could be a very rapid process. So this decrease could be before the desertion. And our results indicate that higher prolactin levels after acute stress in males than in females may be involved in maintenance of parental care. So these higher prolactin levels indicate that the males are more resistant to stress as regards the continuation of parental care. So both the behavioral and hormonal responses of females seems to indicate that they are more sensitive to stressful situations. And the last, males and females face other constraints or trade-offs. I would like to thank all people for their work, for very hard field work, carp farmers for their cooperation. Field work was carried out with appropriate approvals, and this work was supported by National Science Center. And thank you very much for your attention.
Well, thanks very much for that fascinating talk. That's really interesting. Do we have questions? If someone would like to raise their hand in Zoom or potentially just unmute. I see a couple questions in the chat. We'll just start with Liz, who asked, if desertion doesn't have a significant effect on chick fledge rates, are you aware of other costs to males or to the chicks?
The breeding success is very high in this species, and female desertion has no impact on breeding success. It is very difficult to study this term during post-fledging period. So we didn't know, we know nothing about cost for the males, but probably in this species, males and females make the same pairs in the next years, and we have this situation in Whiskered Tern. So probably female desertion could be the benefit for the male, because females have more, we have more energy for the next breeding season.
Thank you. One last, hopefully quick question. How long do males continue post-fledging care, if that's known?
It is a very good question. For at least 1 to 2 months, because we were able, because we are making the birds, so we know it. So for at least 1 to 2 months, but we didn't know nothing about the migration. So maybe also during migration, males feed the fledglings, but we didn't know it.
Well, fascinating species. I guess we'll move on to our next talk, which will continue the tern theme, but shift to the northeastern coast of North America, or the U.S. Northeast. And Alia Caldwell will talk to us about common terns and roseate terns there. Alia, take it away.
All righty. Are we seeing slides?
That's excellent, Alia.
Great. Okay, so hi, I'm Alia, and as Don said, I'm going to be talking about microplastic ingestion by common and roseate terns breeding in the Northwestern Atlantic. So before I start, I just want to mention all of the wonderful collaborators and funders who have made this project possible over the past few years. I can't realistically go through all of these people and funders, but we are so grateful to them for their support. I do, however, want to mention Brian Pelestis, who we miss dearly and whose contribution to tern conservation and science is and will remain deeply appreciated. So, microplastics are ubiquitous in marine systems at this point, and their concentrations are really only expected to increase under current management conditions. So microfibers are one of the most common microplastic forms, and estimates state that about 2 million tons of these fibers enter the oceans annually. And these microplastics enter the oceans via a variety of routes and from a variety of sources. They can be inputted into marine systems either as microplastics initially, so with an example being like from the cosmetics industry, or they can be inputted as macroplastics, which then break down into smaller and smaller microplastics. And once these plastics are in the system, they become available for ingestion by marine organisms all along the food chain. And ingestion of these particles has been documented in all major marine systems investigated at this point. And the ingestion of these microplastics can have a number of detrimental impacts to organisms. These include things like tissue damage, disruption of growth or development, and also the leaching of potentially toxic chemicals into body tissue, which can then in turn disrupt endocrine activity and other systems. I think it's also worth mentioning or noting that humans do have the potential to be impacted by these microplastics as well, as we ingest marine organisms and can gather tissues from their tissue or gather plastics from their tissues. And microplastics have been found in human feces, so this is another thing to be slightly concerned about potentially. So while we do have a somewhat well-established framework for how microplastics impact organisms and processes in the marine system, the field of microplastics research is still really new. And so we have lots of questions in each of these areas, and also just across taxa, and especially within certain systems, questions really do remain. So like other marine organisms, seabirds are exposed to microplastics in their environment, and they're exposed both via primary ingestion of plastics from the water column and secondary ingestion of plastics through their prey items that may have ingested plastics. I'm sure many of you have heard this stat, probably from me, to be honest, but Wilcox et al.'s estimation that 99% of seabird species and 95% of individuals will be impacted by plastics by 2050 continues to be concerning. So in light of this, and because seabirds tend to be particularly sensitive to these kinds of anthropogenic pressures, there really is significant concern over their interaction with microplastics. And this sensitivity of seabirds also tends to make the group useful as bioindicators for pollutants. So this is something that's been well documented in heavy metals, for example. And this idea has been explored a bit and mostly been explored in macroplastics. So there is growing interest for the potential use of seabirds for also monitoring smaller microplastics in the environment. All of this being said, like in most other systems, many species of seabirds still do remain underdescribed or entirely undescribed when it comes to both the pollutant in general, but then especially when it comes to microplastics and even more so microfibers. So the fibrous microplastics that make up the majority of microplastics in the marine environment. So terns are one of these understudied groups of seabirds, and this is despite significant concern over the conservation of many species of tern. There's been some research on macroplastic in terns, but very little on microplastic and really nothing that I'm aware of that directly investigates microfibers. And this is despite the fact that terns likely ingest more plastics within the micro range than the macro range based on their body size and the fact that they likely ingest plastics via their prey items, which can in turn only accommodate smaller plastics because of their own body size limitations. So things like gape limitation. So our study aimed to fill some of these knowledge gaps by assessing plastic ingestion in common and roseate terns in the Northwestern Atlantic. To my knowledge, there's really no research specifically addressing microplastic ingestion by common terns along the Northeastern U.S. coast, and roseate terns remain entirely undescribed globally. So we aim to quantify sort of a baseline understanding of microplastic ingestion in these two species, while also gaining some understanding of the dynamics and mechanisms of microplastic ingestion in the system. So specifically, we were interested in investigating any age-driven differences in microplastic ingestion, any regional differences, and then lastly, any interspecific differences between the two species. So for our first question, we predicted that adults and chicks would differ in their ingestion rate and size. This is because we assume that microplastics are likely being ingested by terns via their prey items, and adult and chick terns do tend to ingest slightly different diets. And then because microplastic size tends to correlate with body size, we also thought that adults would ingest larger plastics. We expected to see spatial differences in microplastic ingestion by terns as well, because microplastic pollution tends to differ regionally. This is a result of things like ocean currents, proximity to land, anthropogenic use of the area, and other factors. We did not, however, expect to see plastic size differ between the regions. And then lastly, again, due to dietary differences between the two species, we expected that common and roseate terns would experience differing rates of microplastic ingestion, but because they eat fishes of similar size, we expected that the size of plastics that they ate would be similar. So we had 3 sites at which we collected fecal samples back in 2019. And common tern adult feces was collected from all 3 of the sites. Common tern chick feces was collected only from New Hampshire and New Jersey. And then roseate tern chick feces was collected in New Hampshire alone. So to isolate plastics from the fecal samples, we first collected a dry weight. Then the samples were chemically digested to remove any biological materials. They were vacuum filtered onto 2-micron paper filters, which were then inspected under the scope for suspected microplastics, which were then removed, imaged, measured, and then they were stored for further analysis that I'll talk about in a couple slides. So this is just an example of what those suspected plastics looked like under the scope. And during this step of the process, we found that almost all of our suspected microplastics were fibers, so something like 85% or something like that, which was expected because they are the most ubiquitous plastic form. So for the rest of our study, we focused primarily on microfibers. So you can assume that everything that I'm talking about moving forward is in relation to microfibers. So to control for contamination throughout the process, we only collected fecal samples from substrates that were non-synthetic, so rocks, skin, cotton, stuff like that. We collected field and lab blanks throughout the process, and then we conducted all of our lab work under a fume hood or other covering to reduce airborne contamination. So a subset of our suspected microplastics were sent away for spectral analysis via FTIR and ATR. So this top image is a spectral reading of one of our particles, which can then be identified by comparison with two spectral libraries. So that's the two spectra underneath ours are the matches, the two top matches for our spectra. And then based on that raw data, the particles were further categorized as either synthetic, semi-synthetic, dyed natural, or natural. So the natural fibers are organically derived fibers, and then the rest of those categories are anthropogenically derived. So here we just have a really quick summary of some of our general results from our characterization of our microfibers. The table you're seeing is just an example of what the raw data look like. So we found that about 18% of our fibers were synthetic, around 1% were semi-synthetic, 45.5%-ish were dyed natural, and then around 36% were natural. So altogether, about 64.2% of our fibers were anthropogenically derived. And those fibers tended to be blue, but some other well-represented colors were transparent, gray, and pink. So moving forward, you'll see results comparing estimations of synthetic fiber numbers per gram of feces between groups. So we estimated those based on our total counts of suspected fibers and these results from the FTIR. So here we have a box plot with the number of microplastic fibers per gram of feces on the y-axis, and then you'll see that dotted red line, that's just denoting our background contamination level. And as you can see, common tern adults and chicks ingested similar microplastic loads. And then here we have a plot showing the size distribution of particles, and you can see that adults ingested significantly longer microfibers than adults did, or than chicks did, and then they also ingested a broader size range. So to revisit our predictions, we can see that our results here were partially supported for the first, or our results partially supported our predictions for our first question. So here we have another box plot, this time showing plastic ingestion across the 3 states that we sampled from. So we found that common terns in Massachusetts ingested significantly more fibers or microplastic fibers than those in New Hampshire, with no other pairwise differences being observed. And then as expected, the microfiber length across the 3 states did not differ at all. So again, here we see that our predictions were partially supported, as we found some distinction between sites, but we didn't see that trend hold all the way across the board. And then lastly, we have another box plot, again, this time looking at common terns and roseate terns. And here we see that the 2 species differed significantly in their plastic ingestion, with roseate terns having higher microplastic loads than common terns. And microfiber length did not differ between the 2 species. So here we see our predictions supported. So these differences that we observed in microplastic ingestion between the species and across regions, and then also the lack of differences that we saw between adults and chicks, can potentially be described by diet. So while we initially thought that adult and chick common terns might differ in their microplastic load due to dietary differences, the 2 age classes do actually ingest a similar diversity of fishes, though in somewhat different proportions. Therefore, it's possible that their diets were weren't actually different enough to produce a signal when it comes to microplastic ingestion load. That being said, the 2 did differ in the size of plastics ingested, which might be a result of differences in the prey items ingested, as we might expect that adult terns can accommodate larger fish, which might in turn be able to accommodate larger plastics. So that might explain the difference in plastic size. In thinking about the spatial results and the fact that Massachusetts had elevated microplastic load, we might again consider diet here as an explanation. And we might actually start to look towards sand lance as a possible route of exposure because common terns in Massachusetts specialize quite heavily on sand lance compared to other regions. And further support for the potential linkage between sand lance and microfiber ingestion rate lays in the fact that roseate terns, which also specialize quite heavily on sand lance, had elevated plastic levels when compared to common terns. While there's no research in the region on plastic load in sand lance, or really in any of the prey items ingested by terns, there is a bit of work in the Pacific that does suggest that sand lance experience elevated microfiber loads. And this kind of makes sense. Sand lance are utilizing the bottom substrate, and there is thought that bottom substrate might be more likely to catch microfibers and therefore have higher concentrations. So if sand lance are using that habitat, it's possible they have elevated microfiber loads. So maybe sand lance are the evil culprit here. No, I'm kidding. We all love sand lance. So if we want to try to take some of the heat off of sand lance and off of diet, the other possible explanation for the differences that we see is related to the ingestion of seawater. So in this scenario, the major microplastic exposure route would be via primary ingestion of seawater. And any differences in microplastics observed would be a reflection of those present in the surface water that birds interact with. So this explanation is certainly plausible, but I think it's less well supported by our results. If this were the case, we would have expected to see really strong differences in microplastic ingestion between adults and chicks. And this is because they vary a large amount in their interaction with seawater during the pre-fledged time period when these samples were taken. If this were the case, we also might not expect to see the differences that we did see, or we might not expect to see such strong differences between common and roseate terns, as the two do tend to use similar waters. They're breeding together on the same colony, probably foraging in similar areas. So to clarify all of the speculations I just threw at you, we are hoping to get around to analyzing prey items for microplastics sometime in the near future, along with water samples from the areas surrounding the colonies and at foraging locations. Eventually, we also hope to assess whether the ingestion of these plastics that we've observed actually have any physical impacts to terns. And so I think there are sort of two major take-home messages from this study. The first being that terns actually might be a potential indicator for microplastics. So the fact that we were able to parse apart differences in plastic ingestion across space within the same species is promising for their use. But the next step is to really figure out how microplastic ingestion levels in terns are quantitatively related to those in their prey items and those in the water column. And then lastly, diet may be the primary driver of plastic exposure in terns. So with that, thank you for listening. I'm going to leave you with this beautiful picture that Gemma took, and I'm happy to take any questions.
Thanks, Alia. There are a couple comments in the chat I'd refer to you, and a question. David asks or comments that he and others have found both fibers and microfragments in the GI tracts of royal terns in South Florida. Do you have any indication about GI tract passage times or differential retention between fragments and fibers?
Okay, differential retention between fragments and fibers. In terms of GI tract, like you're asking how long the feces like is hanging out. I'm not sure about that. That actually might be a question that Gemma could answer because she's probably been thinking about that a lot more, thinking about fecal DNA and like when feces is indicative of diet. I also don't know much about retention time between fragments and fibers. I could speculate, but I'm going to try not to.
Great. Well, I think we'll leave it there in the interest of time, but I'll You might look through the chat and answer some other questions virtually.
Yeah, happy
But
to.
great. Now we'll move on and we'll change taxa from terns to rails and stay in North America. And we're excited to have Lisa Elizondo present to us on some work both on the Atlantic and Gulf Coast. So Lisa, take it away.
All right, let's see if I can get this to present correctly. Does that look good?
I'm still, there we go. Now we have it in your whole slide. Excellent.
Thank you for that introduction. As you mentioned, my name is Lisa Elizondo. I'm a PhD candidate at the University of Delaware. I'll be talking about some of my dissertation research today on the population genomics and hybridization between king and clapper rail. So I'd like to start by acknowledging the many, many people that have made this work possible. As you'll see in a minute, I do have samples from a reasonably large range across the US, and it wouldn't have been possible without the help of all of these collaborators. So king rail, Rallus elegans, and clapper rail, Rallus crepitans, are sister taxa which hybridize in brackish areas. So this is a brackish area in Delaware, and you can see that we do have a king rail looking bird on the left and a clapper rail looking bird on the right. For those of you that aren't familiar with the field marks, this sort of starker contrast up here versus the clapper rail, as well as the darker lines in the barring on the side, are a couple of the indicators, but the obvious one is this bright red breast versus more of a washed out look on the clappers. There's a lot of variation in clapper rail though, and they do get quite a lot darker than that, with dark extending down into the breast as well. So king rail are predominantly a freshwater species, while clapper rail are predominantly in saltwater. As I mentioned, they do hybridize in these brackish marshes. With sea level rise and continued habitat loss and pressures, we're likely going to be seeing more secondary contact as we move forward. So it's important to understand the extent and direction of hybridization between these two species. King rail is broken down into two subspecies, the ones that are in the US and then the ones in Cuba. Clapper rail, on the other hand, are broken down quite a bit more. In Delaware, I have Rallus crepitans, which is where the bulk of my banding research is. And these are a migratory subspecies, whereas some of these southern birds are residents. As you can see, even though king rail are the freshwater taxa, they do still have somewhat of a coastal distribution, just varying depending on where they can find marshes to subsist in. So my objectives going into this were to determine the population structure of clapper rail across their US range, considering that we have all those different subspecies, just to sort of get an idea of how much gene flow there is between these populations. I also wanted to the best of my abilities, evaluate the extent of introgression between king and clapper rail in the clapper populations that I've been sampling. To that end, we collected blood and tissue samples from either banded or collected individuals. We used dd-rad-seq protocols and paired-end Illumina sequencing in order to produce our reads, which I then processed with the Stack's reference aligned genome against our clapper rail reference genome. Ultimately, after filtering, we ended up with about 5,300 SNPs to work with, and so that's the foundation of the downstream analyses that I'm going to be showing you. To sort of try and get a hold on some of these differences, I ran a PCA for just the king rail, just the clapper, and both species. These were just subsetted out of the main overall filtered data set. They weren't refiltered or anything. I then used program structure on the full data set containing both species in order to try and cluster out whether or not king and clapper rail and their different groups would parse out in program structure. And then I used package introgress in R to assign hybrid classes. I used parental references from pure king rail in North Carolina and pure clapper rail in Delaware. For our clapper rail sampling, I was able to get samples from Connecticut down to Georgia along the East Coast, and then for the Gulf Coast, Louisiana State University provided samples from clapper rail there. LSU also provided known hybrid samples as well as king rail samples. And the majority of my pure king rail samples came from North Carolina and Sue McRae's work out of East Carolina University. So for our king rail only PCA, as you can see, the Louisiana and North Carolina birds didn't actually cluster differently at all, which was a bit of a surprise given how far apart they are geographically and how disjunct some of the king rail populations have become with the loss of freshwater marshes. When we look at the amount of variation explained by these principal components, we can see that there's not a huge difference between PC1 and the other principal components in here. So it's probably not a massive driver of this variation. For the clap rail only PCA, we do see that a lot of the Louisiana rails are clustering over here to the side. There are also some of these dark blue Delaware birds interspersed throughout here. And so just as a reminder, these are birds that were identified in hand and banded. So we could potentially have hybrids in these clapper rail depending on how they were identified in hand. Again, though, we don't see a huge amount of variance explained along PC1. It's just above 1.5%. When we run everything together, though, we do see the king rail clustering out from the clapper rail. So obviously I had a clapper rail heavy sample, so you can see all of these ones clustered over here to the left. And so left to right, we seem to be seeing clapper rail on the left and king rail on the right. In the center here, we have some of these known hybrids from Louisiana, these orange dots. We also have a few of our Delaware birds, which, based on this, are most likely hybrids. And then on PC2, we also have some of our North Carolina king rails separating out from the other ones. But if you take a look at the amount of variation explained, the overwhelming amount is explained by PC1, which is over 10%. And so PC2, where those North Carolina birds are split out, is probably not super important. Moving on to the program structure analysis. So program structure indicated that there were two populations in the full data set that we gave it, which presumably are king and clapper rail. And so you can see this lighter blue color seems to be clustering the king rail together, and this darker blue is clustering the clappers. So as a reminder, for those of you who maybe don't look at program structure plots all day, every day, each of these lines is an individual bird that was sampled. And the different colors are the probability of assignment to either one population or the other. So these ones that have, that are getting around 0.5, that means that they have a 50-50 chance of being king or clapper rail. So to sort of get at actually classifying the hybrids themselves, I did use package introgress. And you can see we did have 4 F1s that arose in the center here out of our Delaware birds, which are these dark blue dots. We also had an F2 individual in Delaware and an apparent F2 in the North Carolina king rails as well. So to take a look back at that structure plot and break it down a little bit more, so I've sort of tilted it to the side. Those F1s and that F2 in Delaware, so these are only Delaware birds here, they do line up to those program structure bars that seem to be indicating F1s. So it's a pretty good indication that, this blue is king rail and this darker blue is clapper. As you see, though, there is some odds and ends blue, light blue color interspersed throughout here. And if you look at the hybrid index, you can see that there were another 10 individuals that probably had some level of introgression, either in recent history or further back. Which that means that only 75% of the birds that we banded in hand as clapper rail were pure clapper rail. And just to demonstrate that it's not because I'm horrible at identifying rails, I want to show you guys these 4 photos and let you take a guess at what you think is a king rail or a clapper rail based on those key identification points that I showed you way long ago in the beginning of the talk. So I'll give you guys just a minute on that. So we have a king rail photo that I borrowed from the internet up here in the top right. And you can see it does have the warmer colored face that you would expect to see with a king rail. Our typical clapper down here in the bottom left has the very washed out gray color. And then the bottom right, this is actually an F1 hybrid, which has the really classic dark gray clapper rail face and dark gray down into the breast as well. In the top left, we have something that in hand I suspected may have been a hybrid. And it's actually one of those ones from the group of individuals that just had some amount of introgression. So this is not an F1 or an F2 individual. So moving forward, I do have just a few more samples to incorporate that I collected in 2021. I've done the lab work on those, but we need to get them sequenced and analyzed. And I would like to control for any potential relatedness between the sampled individuals. We did, of course, work on getting individuals that weren't from exactly the same place to incorporate, but especially the banded individuals, we could have some relatedness at play there. I also want to further quantify hybridization across the full data set using a DAPC, so a discriminant analysis of principal components. And I am just now starting some lab work on whole genome sequencing of some king rail and some clapper rail. And from there, I would like to do a genome-wide association study. So that'll just be looking along the whole genome and seeing where between these two species they're actually differing. And with that, I will take any questions.
Lisa, thanks so much for that talk. It's really neat to see hybridization so well characterized as in your work, characterized genetically. We have plenty of time for questions. I see we have one from the chat. Is this the first study to compare the genetics of these two rail species?
So there was one previous study with clapper. Well, okay. So the short answer is no, but this is the first next generation sequencing look at these species. So there was some work fairly recently using microsats, but they used predominantly hunter collected samples, and the hunting seasons correlate with the migration of these birds. So potentially there could have been a lot of migrants in their samples. Whereas we sampled in August, like right after they had finished, but before they migrated.
I would just ask a question. You had some confirmed hybrids from Louisiana. I'm just curious how they were confirmed as hybrids.
Yeah, so they were identified in hand. So James Maley did his dissertation on Rallus rails, and they did find some characteristics that were useful for differentiating them, and he did do some mitochondrial DNA work with them because he was looking at parsing out the different species of rails. So that's when Ridgeway rail got split off from clapper rail. So those were identified in hand, and some of them I don't think are right. If you look at the program structure plot, you can see that some of them appear to have been misclassified. Which is not surprising.
I guess that was my real question. Thanks. I see Gail has her hand up. Gail, do you want to ask a question?
Yeah, very interesting talk. Thank you. And I have two questions, but I'll just ask the one that's maybe doesn't display my ignorance of population genomics quite so much. And that is there conservation implications from your study?
Yes, so king rail in a lot of places are threatened. Freshwater marshes have obviously taken a huge hit, as everyone here probably knows. So it sort of seems that, just anecdotally, because I haven't been able to quantify this yet, but it sort of seems like the direction of introgression is favoring clapper rail. So this could potentially pose a risk to king rail populations as, you know, salinity is encroaching further into these areas. It also, you know, sort of comes into play when you're thinking about what's a pure king rail population and what should we be conserving for this species. And then you said you had a second question?
Well, it was the question, it's your final comment about moving forward and looking at the relatedness of the F1 generations, because one of my questions was maybe the F1 generations can only mate with each other. Like, do you know if the hybrid has problems getting mates and then ends up only mating with other hybrids, and therefore you're going to find some more relatedness amongst that grouping?
Yeah, so it doesn't seem that there is a big bias there. Obviously, rails are not the easiest to observe, to go out and just watch and see what's going on. But there's definitely enough back crossing going on in these populations that, like, I think it's pretty likely that there's not a big disadvantage to being a hybrid. And I know so I work with the Salt Marsh Habitat Avian Research Program. And I know in sparrows, they've actually found the Nelson sparrow introgressing into salt marsh sparrow have a benefit from that hybridization because they gain some ability to tolerate salt better. So it's possible that those hybrids, are going to be potentially even more apt to be mated with in those brackish areas than pure king rail. If that makes any sense.
One last question from the chat. Do you have plans to use the full sequencing result to do any disease ecology study?
I do not, no. And then I see also, based on the sample size, do I expect the cost to be very high? So we're only doing 30 birds. We're doing 15 pure king rail and 15 pure clappers, which were selected based on where they were collected and then also checked against the program structure plot just in case. But I'm hoping that once we sort of get our footing with looking at the differences between the species, we'll be able to move into sequencing hybrids as well and seeing if we do have differential introgression going on there.
Great. Well, thanks very much, Lisa. This certainly generated some great questions. Great talk.
Thank you.
We'll move on now to our next speaker, and we'll take a geographic jump from North America to Southern India. And we have Akshita Mahapatra, I apologize for my poor pronunciation, who is going to speak with us today about herons in Southern India and both spatial and temporal nesting patterns. So Akshita, take it away.
Hi there, can you hear me?
We can hear you well. Would you like to share your slide?
Yeah, just a second.
Okay, we see your slide in edit mode. There you go. That's
Is it in
perfect.
full screen?
That is perfect. Please
Okay,
proceed.
thanks. Okay. Hi everyone, greetings to all. I'm Akshita. I'm a PhD student, mainly researching on colonial nesting water birds from India. So today I will be presenting part of this research study that I have been doing from last 3 years. And this is for a particular location, a particular heronry that I'll be presenting this work. And in this heronry, these are the two dominant species which nest, painted stork and pelican, which are also globally near threatened. These are the dominant ones. So the heronry that I am referring is very unique. It's called kokore bellur. So in local language, it means kokore means stalks and bellur means home. So it's like abode of stalks. And it's unique as it's situated far inland from the coastal regions and in midst of dry plantation landscape. And especially this location, it's inside a very small village of just 3.12 square kilometer and with no immediate water source. Also, it's known to be a very traditional breeding site for these birds for more than 300 years now. And maybe even more, since the village was established just 300 years ago, so that existence is known to us since then. But most likely it's existing from a long, long time. And another unique characteristics of this place is the special bond that exists between villagers and birds. It's for villagers, it's a lot of emotional connect to these birds. They treat them as their own daughter as they come back every year for nesting, for delivering their chicks and raising them. And it's a very common traditional tradition that is also followed in this human society in India. And also same time, it is also believed that birds also nest very close to these villages as they feel more safer. Because there are similar kind of trees which are there just adjacent to these villages, large patches, but they don't go and nest there. They prefer to nest very close to those trees which are just next to these the built-up areas of these people. So the mixed heron rate consists of pelican painted stock and also black crown night heron, little egret, and little cormorant. And a major objective of this study is to understand the spatial and temporal nesting patterns of this mixed species heronry. So for a temporal nesting pattern, this data that I'll be presenting here is of the last two nesting seasons, 2018 to 19 and 2019 to 20. For the temporal variation, we were seeing the arrival time of each species and when they are initiating their nesting, and also directly counting all the nests. So it's very small area, so I'm able to count each individual nest of all the species. And for the spatial variations, we are seeing their nesting tree preferences and the tree characteristics of nesting trees as well as non-nesting trees. And once all the nests are settled, we then go for collecting the nest characteristics like nest height, placement, their inter-nest distances, all these were recorded. So first I'll be sharing nesting periodicity of individual species of the heronry. So pelicans, they arrive first to this place in mid of October, which also coincides with the receding northeast monsoon in India. And another interesting point to note here is that we are seeing that there are these two populations which are coming. First major population comes in the month of October and they keep on coming till December. Then there is a gap. And then there is a smaller flock of around 20 to 30 individuals which come in the month of February. So it might be same individuals who have probably failed in the first attempt for nesting. They are trying to do it in the second go, or else it might be total different population. But why they are coming and waiting till February to begin nesting is an interesting to look for. Next, which comes for nesting are painted stock. They arrive here in the late January, and they keep on coming till March or April. It's in one go they'll come. There is no such similar to pelican. It's a unimodal nesting population, and they choose to come in this dry period when there is almost no rain. And if we remember from pelican, the second nesting which starts, it coincides with the arrival of painted stock. So we see that a majority of the painted stock, they start their nesting in February month, and at the same time pelicans also begin their nesting. For night heron and also other water birds, they wait for painted stock to begin their nesting. And we have seen a huge increase in their population. If we see this in 2019 to compared to 2020, the population has increased from 101 to almost 560 nests. So also at the same time, we saw that they are also nesting second time in the month of June, which is going till August. Similar patterns were also seen in case of cormorants and egrets. They were not in very large number as compared to painted stock and night heron, but still they also followed the same nesting pattern. In the second season, there was a huge hike in their nesting population as well, and they also continued to nest till August. So before going to the spatial variation, how they are distributed inside the village, I would like just to explain about the nesting habitat of the village. So dominant trees which we find here are Thespacia, Delonix, tamarind, ficus trees. These are actually long back planted trees, maybe from the starting of this village when it was established. So these are all human-made habitat. It's all plantation which they did back then, and it's in those trees they are coming and nesting. So if we see the availability versus use of these trees for the most dominant ones, so we can clearly see that they are selecting almost majority of the available trees, whereas there are some species or same trees which they are not preferring to nest, the red ones. So if we see the characteristics of nesting versus non-nesting trees, it gets clear that non-nesting trees are younger trees, as they have in the second table, right corner, top right corner, we see that there are the nesting trees, they are having less GBH as compared to non-nesting trees, and also canopy spread is also less. So probably these are not suitable to support such densities of the mixed heronry species. So when we see the nesting tree preference of each individual, pelicans were found to be very specialist, which nest mainly on the ficus trees. We can see that it's almost 80% of them are coming and nesting here, and which are also in the periphery of the village, not in the middle tree patches, it's in the periphery of the village. And pelican also segregate from the rest of heronry in both ways. Temporally, we also saw that they are coming first in the month of October, and also spatially, we are seeing that they are going and establishing their own species-specific colony inside the heronry. However, if we see this blue bars and gray bars are the second clutch which are nesting in February month. If we see this, they are preferring, their preference is for different type of tree species, and which also are, we have seen that painted stocks were already nesting there, and they are going and nesting in those places, so which is inside the village. So we are seeing that initial flock, they are very particular. They are only going and nesting on the ficus trees, whereas the second flock, probably because they want to nest together, so they are going and nesting along with these other species. In contrast to pelicans, painted stock seem to be more generalist, and they nested in all types of trees. But majorly, if we see that they are nesting in this Spatial, which is also the most dominant tree which is present in the village. But we have seen that they nest in even very 10 feet height to 30 feet height, or very young trees to very old trees. So they seem to be very generalist. Night heron showed very similar preference as that to painted stock. All they nested 100% in those trees where already painted stock nests were there, and the nesting preference is very similar to that of painted stock. Whereas for cormorant and also egret, they mainly preferred one type of Dillonix trees primarily for nesting. And but we also saw that cormorant and egret, both, if we see, both of them are preferring this Dillonix tree more, and both of them also nested along with painted stock where they were nesting. Since cormorant and egret were very few in number as compared to painted stock and night heron population, so we focused more towards the association that we were seeing in case of painted stock and night heron. So we went ahead and checked how the nests are placed inside those trees, how they are segregating. So first we checked this mean nest height of both of the species, and we found that there is not much difference. All of them in all these different tree species, they are nesting together only in same height. Only difference was seen in the tamarind tree. That is probably because the nest density of painted stock and tamarind trees was very high. It was almost 30 to 40 nests in single tree. So might be the competition for night heron to place their nest next to them would be difficult. So they are nesting a little lower. And when we checked how close they are nesting to the painted stalks, night herons, it was found that they were nesting almost from negligible gap between painted stock and night heron nest to a gap of just 1 meter. So it was very close. So these are just preliminary summary data that we are now in process to analyze it in more detail, mainly to understand what factors are associated to the nesting sociality in colonial water birds, and does close interspecies association lead to higher breeding success if they are nesting very close, and if in some trees they are nesting at maintaining some gap. So does that mean that there is also a difference in the breeding success? And does higher breeder density increases their breeding success? So these are some of the main points we are now looking into for detail and analysis of the data. I would like to thank Karnataka State Forest Department for funding this study and providing the support and field, to all the members of Waterbird Society for giving this opportunity to share this work in this platform, and especially to all of you for joining me here. Thank you so much.
Thank you for a great talk, Akshita. There's some comments in the chat. Maybe we'll start with the question from there. You said that there is not much water around, particularly during a portion of the nesting season. Do you know how far these birds are traveling to forage?
That is still a mystery. We don't know from where they are coming. In this season, just maybe in January month, we will acquire the tags and we will be putting to these pelicans to know exactly from where they are coming and how much distance they are traveling. But it's still unknown to us. In its entire range, it has never been tagged. So this will be the first pilot study.
Wow, that's some great follow-up work. Another question, given the rich human culture around this colony that you described, do you think people of the village provide food for the nesting waterbirds that may contribute to their population increase?
If they can provide food, is that the question?
Yeah, do you think people help by providing food in some way?
Actually, we are in process of tagging these birds. We are planning to bait them in a small pond that is being artificially created, but we don't know whether it will be successful or not. And if I would like to answer your question of the role of these villages to help their population here, so they have been since 1995, they have been rescuing the fallen orphan chicks. Usually every season there are lots of chicks that fall from their nest, and usually these feral dogs, they will eat them. So before they go and hunt them, they rescue them, and there is now forest department is actively involved in supporting these things. And we are in every season, we are last season there were 120 rescued chicks that were successfully fledged after taking care of them for 3 to 4 months. So that is a huge step that these villages are taking in for the protection of this population.
Wow, that's really nice. I guess I have another question. Are there any predators that can get to the nests in the trees?
So I have been seeing these Brahmani kites are one particular species that are the prey, and that we usually find them nearby, and also sometimes we have seen some kind of egg predation, but more or less we have seen them just sitting and waiting for these pelicans or other species to drop fish, and then they will go just down to collect that, not actually any conflict with the active nests. There were very rare instances of them predating on the nest for egg or so.
Another question, just this relationship between the villagers and the birds is really fascinating. Do the villagers use the trees for anything other than shade? And is there any concern about the birds perhaps killing the trees due to their guano? Cormorants certainly are well known for killing trees that they nest in.
Right. Yeah, right. So this has been a concern from last few years actually, with this growing development, commercialization and everything. So now, since before they were using those guano, those birds dropping and using it as an fertilizer for their agriculture fields. But now that concept is fading away, and they are more depending on the artificial fertilizers they get from markets. So since the economic benefit that they were before getting is now has been changed, so now the people have started seeing like kind of a nuisance. So they want now to cut down these trees and establish their own property in those areas, because since these all the trees are just adjacent to this villager's household. So that is a concern. They also use these, lop these trees for getting some, they feed these ficus trees and use it as to feed their goats and cattle, and tamarind trees for fruits. But they don't do any harm in the when the breeding season starts. So till 6 months they will not go and touch these trees. But in the non-breeding time, yeah, they do lop the trees sometimes, and there are cases of chopping the whole trees as well.
Wow. Well, I hope the relationship, a positive relationship, continues between the villagers and birds. It's a really heartwarming story. I guess, yeah, thank you so much for the talk. And I guess we'll move on now to the last talk in our session. That'll be given by Bradley Wilkinson, and we'll switch back to North America and the Southeast coast. But we'll continue thinking a bit about pelicans, although a different species here. Brad, go ahead and take it away.
Great, thank you. Well, I want to thank everyone first for attending my presentation. I'm going to be presenting the last chapter of my dissertation, which I just completed at Clemson University in the South Carolina Cooperative Fish and Wildlife Research Unit. So I want to start off by kind of making a general statement that migration is a common adaptive behavior that results typically in increased growth, increased survival, or increased reproduction across space and time for the individuals that choose to undertake migration. However, the drivers of migration are complex. Typically, we see kind of a hierarchy of factors where both intrinsic and extrinsic drivers layer on top of each other in some way as to precipitate a migratory movement. And untangling exactly which one of those factors plays a critical role can be challenging. There's also a situation called partial migration, in where some individuals of a given population will leave and go on a migration, but some will stay relatively sedentary around the same area. And studying partial migration gives us a really good opportunity to test some of those factors and to see which factors specifically relate to some individuals leaving and some individuals going. And importantly, partial migration may also act as an evolutionary precursor to full migration. So if we understand something about the drivers of partial migration, we may understand why full migration has developed evolutionary over time as a whole. My study species is the eastern brown pelican. It's a long-lived apex coastal predator which breeds generally from South Texas to Chesapeake Bay in North America. Forages on surface schooling marine fish. It's a really high-profile, iconic species. The public knows a lot about it, and it's also a species of conservation concern. But importantly, brown pelicans are partial migrants in many parts of its range, including in the South Atlantic Bight, which was my study area. So for those who may be unfamiliar, the South Atlantic Bight is the area of coast which stretches generally from the Cape Fear River in North Carolina to Cape Canaveral, Florida. It's a really dynamic system full of estuaries, salt marshes, and barrier islands with a large number of riverine inputs that influence local oceanography. And so this makes it a really unique and interesting place to study because there's so much freshwater mixing with this really dynamic, like I said, ocean system. So it's a really highly productive area. And there's a ton of seabird colonies in the South Atlantic Bight, which I've illustrated in this figure on the left. So each one of those dots represents the place where seabirds breed in the South Atlantic Bight. But importantly, there's only about 15 pelican colonies annually in the South Atlantic Bight. The largest colonies occur in coastal South Carolina near Charleston, which were the colonies that I focused on for this study and for my dissertation as a whole. But I want to stress the point that this is a very productive system that is, like I said, full of seabirds generally. So some pelicans migrate out of the South Atlantic Bight in any given year, and some do not, which, as I've described, is a hallmark of partial migration. There's several competing hypotheses explaining why partial migration might exist. But as I mentioned, identifying the factors for a specific species or populations can be very difficult. So as part of my dissertation, we deployed GPS units on brown pelicans, and specifically with the role or the goal of investigating the role of intrinsic versus extrinsic factors on both migratory initiation and migratory rate for this population specifically. And I wanted to relate some of the factors that I thought might be relevant to existing hypotheses of partial migration, like I said, so that hopefully we could learn something about the development of maybe migratory behavior as a whole. So on this right-hand image, I just want to highlight the leading pelican there has one of our GPS units on it. We were very fortunate in the South Atlantic Bight that there are a lot of people taking pictures of pelicans, and so we were able to get some great photos. And I just wanted to especially shout out the photographer who allows me to use this image because I really like it. Okay, so some of the hypotheses that I thought might be relevant. I went through the literature of partial migration, and there's probably a dozen or so hypotheses that could be explanatory for partial migration, but I've chosen just 3 to highlight here that I thought might be really important for our system specifically, kind of a priori. And so the first is the competitive release hypothesis. And this hypothesis generally states that dominant individuals will tend to remain in place, whereas subordinate individuals will tend to migrate. And this could be because dominant individuals might be larger, they might be more competitive from a foraging standpoint. But regardless, like I said, the big tenet is that if you're a dominant individual, you can afford to stay put. But if you're a subordinate individual, it would be better for you to migrate to somewhere where there's less competition for access to resources. And importantly, the competitive release hypothesis is a density dependent response. And so you can imagine a situation where if you're only competing against 100 other pelicans, even if you're not a strong competitor, you might be able to remain in place. But if all of a sudden there's 1,000 pelicans, you might have to be very competitive to remain. And so you might end up migrating even though you're not necessarily that weak of a competitor. And so like I said, it's important to understand that competitive release should be density dependent. The second hypothesis there that I've marked down is the is the fasting endurance hypothesis. And the fasting endurance hypothesis states that individuals which are able to withstand reduced foraging will remain, whereas those at risk of starvation will migrate. And so the fasting endurance hypothesis is really tied to how much of a resource is given in an area. And the thing to understand, I guess, about fasting endurance hypothesis is that it should coincide with some sort of reduction in foraging opportunity or some sort of reduction in foraging availability. So if all of a sudden there's a situation that develops where what was plentiful is now very scarce, those individuals that are able to withstand the scarcity should remain in place, but those who might not be able to gather resources effectively anymore will migrate. And the last one is the thermal tolerance hypothesis. And the thermal tolerance hypothesis is very simple. It states that those that are unable to cope with thermal extremes will migrate. This could be very hot temperatures. This could be very cold temperatures. This could also apply to a population of individuals that are at the very limit of the range of the species. But regardless, it's all tied to, like I said, thermal extremes. And so you could think of a situation for like pelicans where if the ambient air becomes very cold, pelicans might not be able to deal with cold ambient air temperatures, and so would migrate. And so these are, like I said, 3 hypotheses of the several that exist that we thought might be relevant specifically for brown pelicans in the South Atlantic Bight. So to investigate this, we deployed a total of 86 GPS satellite transmitters in South Carolina and in Georgia. And then we defined migratory individuals as those which departed the Carolinian marine ecoregion before March 1st of a given year. I should say that these deployments tended to occur in May, June, and July. And so we needed tracked individuals throughout the fall migration and then during the winter time as well. And I'll speak a little bit more about why we chose the Carolinian marine ecoregion and also what those boundaries looks like on the next slide. So for those individuals which did depart the Carolinian marine ecoregion, we used some functions in the R package migrate R to identify the specific initiation date of when migration occurred. And we did this by comparing resident versus dispersal models. We chose dispersal models because we were only interested in looking at individuals who migrated away from the breeding area and not necessarily returned. So for the full migrant model, you also have to have a return movement, which we were not did not include as part of the study. But like I said, that package allowed us to specify the date, not necessarily on which the bird crossed out of the marine ecoregion, but the date that it began migratory movements. So it's important to keep in mind. So this is a map of the Carolinian marine ecoregion. On the left-hand side, you're looking at our individuals which departed from this marine ecoregion and we defined as migrants. And on the right-hand side, you're looking at a map of the individuals that remained within the marine ecoregion and we classified as residents. And then in the bottom right, you can see a map of the study colonies that we used. I won't go into detail which ones they are, but they were focused around Charleston, South Carolina, and then Brunswick, Georgia. So you can see that the Carolinian marine ecoregion is not necessarily small. It actually encompasses the borders of the South Atlantic Bight fairly well. We chose to use the borders of this marine ecoregion for a couple reasons. The first is that it's inherently ecologically meaningful. The areas that are within the marine ecoregion all tend to be similar to one another. And we were really interested in seeing we're really interested in looking at the drivers of pelican migration on a regional scale. And we wanted to understand why pelicans would choose to leave one given environment for another given environment. So you can imagine, for example, like the estuarine marshes around Charleston are not that significantly different from the estuarine marshes around Brunswick, Georgia. but the areas from like Charleston would be very different compared to like the foraging areas in the Florida Keys or in Cuba or even like Mexico. And so we were interested in highlighting what would precipitate a movement from, like I said, the South Atlantic Bight kind of region to a completely novel ecoregion. And that's how we defined our residents versus our migrants. So in order to discriminate which factors might be important in initiating migration for those individuals which did choose to migrate, we used time to event models. Specifically, we used Cox's proportional hazards model. And what this does is it allows you to input a wide variety of factors into a given model. And then this model has one specific event which it looks towards, if that makes sense. That was not a great way to explain it, but you can imagine a whole row of zeros in a spreadsheet, and a zero corresponds to any given day that individual is tracked. So if it has a 0, that day it did not migrate, it chose to remain. And then you have at the end of that time series a 1, which is when the migration was initiated. So you have a whole line of zeros punctuated by this 1 at the end. And what this model does is it looks at the change in all of those different factors through time until you get to that 1. And it basically allows you to discriminate which factors were important from changing that 0 to a 1 over the course of that time scale. So the factors that we thought may be important and also related to hypotheses that we were looking at were air temperature, sea surface temperature, the amount of chlorophyll A, the North Atlantic Oscillation Index, and also the meridional wind component, which is the wind component from north to south. And those were all time variate and spatially matched to each individual pelican's location. We also included some static variables of sex. We developed a body condition index using bill lengths and also mass of the individuals when they were initially deployed with the transmitter. We used size, and then we also used colony size as well to try to get at some of that density dependence, which I mentioned for the one hypothesis there. So moving towards the results, the factors that we found to be important were sea surface temperature, chlorophyll A, the North Atlantic Oscillation Index, and also body condition index. So these were the factors that were highlighted as being significant in the final model for our Cox's proportional hazards. So in any given year, we found about 74% of pelicans are likely to migrate out of the South Atlantic Bight to a different marine ecoregion. We actually came at that estimation 2 ways, and so I feel like it's relatively robust. Pelican migration typically occurred in November and December. So on the figure on the right-hand side, the gray shading at the very bottom is just kind of like a quick density plot of when those migrations occurred. And then the solid line is the probability of migration over time bounded by the 95% confidence interval. You'll see that the confidence interval, 95% confidence interval, really widens towards the end. And that widening is an artifact of the fact that not all pelicans migrated. So you would have some proportion of pelicans tracked through March 1st that were residents the entire time. And that's part of the reason why that confidence interval is so wide. Like I said, factors that were most associated with initiating migration are shown in the table on the left-hand side. The most highly significant of those were sea surface temperature and body condition. And interestingly, we found that migration probability was significantly associated with an increasing in sea surface temperature and a lower body condition index. And so these results aligned the most, I guess these results aligned best with the fasting endurance hypothesis of partial migration, which I'll explain in a second. So it's important to know that the primary prey of pelicans is juvenile Atlantic menhaden. And menhaden have a very interesting life history themselves. They develop in estuaries over the spring and summer, and in the fall, juveniles migrate out of estuaries to the open ocean. So you have this large population of adult menhaden that are offshore in the wintertime when they spawn, and those eggs and larvae get advected up into our estuaries, our estuarine systems in like the late winter, early spring. And then, like I said, in those estuaries, those larval menhaden develop into juveniles. And when they're juveniles is when they're targeted by brown pelicans. But in the fall, those juveniles will migrate back out of the estuaries, back to the open ocean to join that adult population in the late fall, early winter. And at that point, they become unavailable for pelican consumption by and large. Importantly, menhaden emigration is triggered by prolonged periods of low SST. And so it's fairly robust in the literature if you look that when sea surface temperatures stay relatively depressed for a period of like 7 to 10 days, that tends to trigger this ontogenic emigration of menhaden from, like I said, the estuaries to the open ocean. And so what we saw when we plotted pelican tracks over time as a function of SST, we saw that pelicans tended to initiate migration after a period of low SST when sea surface temperatures began to then rise again. And so on the plot on the left-hand side, it's a little confusing, but each gray line represents an individual migratory pelican that we tracked, and the dot is the initiation of migration. So I want to make it clear that that dot is not necessarily when the individual left the Carolinian marine ecoregion, but that dot is when it first initiated migratory movements as discriminated by the migratory function, which I described earlier. So like I said, you can see that overall the sea surface temperatures tended to decline, but you see this kind of high spike in sea surface temperature. And at that point, that pelican tended to migrate. And so this suggests that resources were low after juvenile menhaden emigrated. We're kind of hypothesizing a scenario whereby you have this depression of sea surface temperature, and then you have all of these juvenile menhaden leaving the estuaries for the open ocean. And that might actually be a very good time to be foraging if you're a pelican. You have this large movement of juveniles who may be more exposed than they would normally. But then as those sea surface temperatures begin to rise again, that emigration may end. And all of a sudden you're left with a depression of resources because all those juvenile menhaden that were in your estuaries have now departed for the open ocean. And so since you have this reduction in available forage, combined with our body condition index, it suggests that pelicans who are in poor condition might not be able to withstand that reduction in forage. So if you're a good competitive pelican who has a high body condition, you might be able to continue to forage on what menhaden are available, or you might just be able to withstand, like I said, that reduction in forage. Perhaps you're more competitive. But if you're an individual who's in poorer body condition, you might be less competitive and you might be less able to acquire resources as those resources have become unavailable. I also want to mention that we conducted a post hoc analysis with a 7-day averaged SST lag, and that model was performed worse than the model that I just specified, and also it gave us the same results. So even when SST was averaged over 7 days, we found the same relationship. I'm planning on doing a sensitivity analysis, moving beyond 7 days to maybe 14, 21, and hopefully trying to discriminate if I can find a relationship with decreasing SST where it then pivots to increasing SST to really understand how far back those pelicans are queuing in on that rise in sea surface temperature for their migratory queue. A couple thoughts. Resolving the role of interspecific competition for this system is really difficult. We didn't find a density dependent relationship, unlike Lamb et al. 2017, which did find a significant density dependent relationship on both migratory probability and also the distance migrated by individuals. Like I said, we didn't find that in this study, but it might be because colonies in the South Atlantic Bight act as sort of one coherent metapopulation. So if you recall, all of our colonies were clustered around like Charleston and Brunswick. And for the most part, there's no other colonies in between those two areas. And so those colonies are relatively close together. They're within like the foraging ranges of each other. And so instead of using individual colony as a proxy for density dependence, it may be more appropriate to think of all of the pelicans in the Charleston area and all of the pelicans in the Brunswick area as acting as two separate populations. And so, like I said, we didn't find a density dependent relationship here, but that might be because we were, the scale that we were looking at might be inappropriate for assessing that. Importantly, interactions between BCI or body condition index and the environment were not significant. And so this implies that birds who are in poor condition will migrate even under equal environmental conditions. So even if there were a ton of menhaden around, if you are an individual who's in poor condition, you are more likely to migrate. Like I said, I think ecologically, it makes sense to think about a reduction in forage impacting individuals who are in worse condition more heavily than those who are in better condition. But it's still want to make clear that, like I said, we didn't find an interaction between those two terms necessarily. Lastly, I just want to say that brown pelicans might represent a model species for understanding partial migration as a whole. Lamidal looked at partial migration in the Gulf of Mexico. We looked at partial migration in the Southland Bight. Because pelicans are large, they're easily tracked. You can get a lot of really good information on individual movements, and this might be an avenue, a species to continue studying, I guess, for understanding drivers of migration as a whole. I just want to wrap up by thanking my field assistants as well as all the people that advised me. And I think I might have some time for some questions. Thank you for listening.
Thanks very much, Bradley. Great work. I guess we are at time. I want to just a couple of announcements, and then we could switch to questions because we don't have any more talks in this session. So we'll have plenty of time for questions. But I wanted to draw everyone's attention to a post in the chat that Liz Craig made. We have a little bit of time before lunch. It would be a good time to visit the Waterbirds Conference merchandise link, and also consider bidding for the quilt that Stephanie Jones so artfully produced. Stephanie's a former editor of the journal and an amazing quilter. And proceeds from these activities, particularly the quilt, will go to support student travel for next year's meeting, which will hopefully be in person. And so definitely a good way to get some meeting swag and contribute. And sorry, I erred on the quilt. It's not a bid. You're buying a raffle ticket essentially for only $5. So I think those are the announcements I wanted to make. Now I'd love to thank all of the speakers for the session, and then turn it over to folks who have questions for Bradley. And I guess I'll start with a question I have. Oh, no, I think Pat has a question. Pat, if you want to unmute.
Sure. Yeah, I have a question about, you were saying that if they're in poor condition, they'll migrate anyhow, but that's sort of counterintuitive to what we think, about migration, that they have to be in pretty good shape in order to migrate. So do you have any explanation for that?
Yeah, I think my thought would be that migration is not necessarily that energetically expensive for pelicans. The migrations that we saw are relatively rapid, like they're undertaken in the course of only a few days. It doesn't take that long for a pelican to get from like Brunswick to the Keys, for example. And so I don't think that, I think it's more important for pelicans to get to an area of higher forage. I don't think they're expending a ton of energy necessarily to leave the Southland Bight to get there. I do think that it's valuable for pelicans to stay in like the Charleston estuarine system if possible, because like I said, it's just a very productive area generally. But during those periods of lower resource abundance, it may be more beneficial to be in the Keys or in Cuba or something like that. And like I said, I don't think for pelicans it takes that much energy to undertake that flight.
Do you know if the ones that go farther are in better condition?
Well, I know from like the lamb paper, individuals, I believe, who were smaller, like smaller skeletal size, tended to migrate farther. And so that's not exactly condition, but it implies that individuals who are smaller to begin with ended up migrating farther away from those home colonies in the Gulf. And And so it's not exactly an answer to your question, but I don't personally have any indication on whether those individuals who were in worse condition did migrate farther. But like I said, it seems to be a relationship with like potentially size and migratory distance.
Thank you.
I see a question from Danielle in the chat. For the pelicans who migrate, do they stop along the way to forage, or are they going nonstop to their destination?
Yeah, it's a great question. We tend to see kind of a mix of strategies. Some individuals will really just book it down south, and some individuals do stop for a couple of days up to a week or two somewhere, maybe like in North Florida, before moving south again. I would say that over like the wintering period, there are some individuals who tend to be very faithful to like specific wintering locations throughout the course of like one individual winter. And we have some individuals who seem to kind of like never pick a defined spot for the entire winter. They'll like migrate to South Florida and stay there for a month, and then they'll move to Cuba and stay there for a month, and then come back to like the Tampa area for a month before going back to Charleston. And so it's actually part of the reasons why brown pelicans are fun, but also difficult to get at some of these drivers, is because their sort of flexibility and movements in the autumn and the winter time, especially, they're really not tied to a specific place. And so, yeah, my answer to that would be we see a mix of strategies between nonstop and also stopping along the line.
Another question that is related, do the pelicans that migrate switch to a new food source at their new location?
Yeah, that's also another good question. I don't have like a ton of information on that specifically for pelicans that we tracked like switching, but I would say the diet studies in like the Keys in South Florida indicate that they don't really forage on menhaden to a great extent. They're more foraging on things like spot, other sort of lower latitude fish. And so the studies that were undertaken in South Florida tended to be during the breeding season. So we're kind of using as a proxy that if pelicans are in the Keys, like Southern Florida, they're likely foraging on what pelicans who are resident there all the time are foraging on. But it would be good just to like kind of, drive that point home empirically that there's a food switch. But menhaden in South Florida tend to be those adult offshore spawners. There's not really those large complex estuarine structures in like South Florida that menhaden need for spawning. And so there certainly wouldn't be like juvenile menhaden in the nearshore system in like South Florida.
I'd follow that up with a question I should have occurred to me a while ago, I think. Is it safe to assume that the pelicans that do not migrate continue to feed on menhaden primarily all winter?
That's a good question as well. That's something that's understudied. Pelican diet as a whole is relatively unresolved throughout like the non-breeding period. Most pelican diet studies have been on like chicks during the breeding season, and then adult forage during the breeding season as well. And so there's not a ton of information on like what pelicans are targeting during the non-breeding season. But yeah, that would be something to get at for sure. I would tacitly assume that there are enough juvenile menhaden, because I should preface this statement with not all juvenile menhaden leave during the autumn. There is a subpopulation of juvenile menhaden which take 2 to 3 years to develop. And so it's not like the larvae that were advected in the spring all leave in the fall. There are, like I said, a number of menhaden which will remain a couple of years in those estuarine systems. And so there should be some young age class menhaden in estuaries all year round.
Cool. Let's see. Juliet Lamb is promoting the idea of fecal DNA from pelicans to get at some of these questions, which is a good thought. I'm not seeing any other questions specifically in the chat. I don't know if anyone else has anything. if not, maybe we could release you all. Again, I would steer you all towards the merchandise and the quilt. And I'd like to again thank all of our speakers and my co-moderator for this session, Liz Craig, and all of you for attending and asking really interesting questions. All right, I think that's a wrap. Everybody enjoy the rest of the day and the rest of the whole meeting.
All right, that was great. I'm gonna go ahead and end the meeting for everybody. Good to see you.
Thanks for all your help.
Yes, it was great.